like to introduce Dr. Karin Seidler, who gained her PhD at the University of Vienna and undertook postdoctoral research in nutrigenomics at the Department of Nutritional Sciences, the Faculty of Life Sciences. Karin graduated from CNELM with an MSc, and additionally, Karin studied naturopathic nutrition and naturopathy at the College of Naturopathic Nutrition. She is currently head of student research, a research supervisor, and module leader at CNELM. So Karin's presentation today is on intermittent fasting and cognitive performance, targeting BDNF as a potential strategy to optimize brain health. Welcome, Karin.
Hi, thank you, Indrani. As Indrani already said, my project is on intermittent fasting and cognitive performance, targeting BDNF as potential strategy to optimize brain health. The theme of the project is the population is in general healthy adults, but also adults with cognitive impairment, Alzheimer's or dementia. And the intervention is obviously intermittent fasting, which refers to extended periods of little or no food intake, alternating with ad libitum eating on a recurrent basis. The most common studied form in animals and humans is alternate day fasting or time-restricted feeding, but there are also other options like the 5-2 diet. The mechanisms I'm focusing on are neurogenesis and synaptic plasticity via brain-derived neurotrophic factor binding to its high-affinity receptor tropomyosin kinase B and enhancing long-term potentiation, dendrite and protein synthesis. The measured outcomes are related to memory and learning and comprise a variety of different outcome measures and tests. The scope of the research, so this was a mechanism review, again, performed in 3 stages based on PRISMA guidelines and critical appraisal. This was the title of the original MSc dissertation. And this was based on a hypothesis that intermittent fasting can optimize brain health and cognitive performance and mitigate age-related cognitive decline via BDNF-mediated improvements in synaptic plasticity and neurogenesis. The primary aim was to explore these mechanisms involved in BDNF signaling in the adaptive response to mild nutritional stress, intermittent fasting, with the secondary aim to evaluate the potential use of intermittent fasting as dietary intervention to enhance cognitive performance. This research was not driven by pathology as primary outcome, but rather aimed to understand how enhancing cognitive function can add value to a functional approach to personalized nutrition and healthcare. So these are the objectives. These were basically to investigate the mechanistic role of BDNF, binding to its receptor TRKB and activating 3 signaling pathways, as you can see here. Then to evaluate the induced effects on cognition, learning, and memory. And as a second part, to evaluate the effects of intermittent fasting interventions on these mechanistic and mechanistic markers and outcomes. It also included epidemiological evidence linking BDNF cognition and Alzheimer's disease or cognitive decline. So aging is a major risk factor for neurodegenerative diseases as the functional capabilities of the brain decline. Brain aging is accompanied by impaired adaptive plasticity and resilience, as well as dysregulation of brain homeostasis and neuronal network activity, which all leads to reduced cognitive performance and a higher vulnerability to neurodegenerative diseases and stroke. Excessive calorie consumption and the sedentary lifestyles can accelerate these pathological processes and hence brain aging, as you can see here as well. Bioenergetic challenges such as intermittent fasting or also exercise or cognitive stimulation have shown to promote lifespan and health span by inducing an adaptive stress response. And in addition to the benefits of caloric restriction, intermittent fasting exposes neurons to metabolic stress, which results in the production of proteins involved in synaptic plasticity, neurogenesis, memory, and learning. So BDNF has shown to play a major role in this process and has emerged as a key regulator of brain health. The brain's ability to adapt in response to experiences or environmental stimuli depends on the plasticity of synaptic connections, and the formation and maintenance of these is promoted by activation of synapses. Synaptic plasticity is considered the cellular correlate for multiple cognitive processes, including learning and memory. BDNF not only regulates synaptic plasticity in the hippocampus and cortex, but was also shown to promote neurogenesis, proliferation, and differentiation of neural stem cells into mature neurons. And this ability is essential for learning and memory formation. And this is also maintained throughout life in a subsection of the hippocampus, the dentate gyrus. Aging-related loss of BDNF has been associated with reduced synaptic plasticity, memory and learning, as well as an increased risk of neurodegenerative diseases. And also the BDNF gene polymorphism is associated with reduced cognitive performance in memory-related tasks and executive function, as well as a higher risk of Alzheimer's disease and cognitive impairment. Intermittent fasting has shown to ameliorate such cognitive deficits, partly induced by high-fat diets in different animal models, which was accompanied by increased BDNF levels and also independent of amyloid beta or tau pathologies. So the mechanism review included an in-depth review of mechanisms, but the paper only focused on the interventions because of the limited scope. And for this publication, the review and intervention searches were updated to include the most recent evidence and resulted in 29 reviews and 53 intervention studies, which are the sole focus of this presentation. But to give you some background on the mechanisms, intermittent fasting can upregulate BDNF via brain intrinsic glutamate receptor activation or peripheral signals such as ketones, which activate relevant transcription factors. So by binding to its receptor, which you can see here, DRKB, BDNF activates three major signaling pathways, which in turn promote synaptic plasticity and neurogenesis, as consistently reported in animal and in vitro studies. This enhanced synaptic plasticity was represented by increased long-term potentiation, neuroid outgrowth, and then dendritic spine density, as well as increased synaptic protein synthesis. A self-amplification system is required to ensure adequate signal strength, which is important in structural and functional plasticity. And this is via this CRAP here. Numerous in vivo studies demonstrated BDNF-mediated benefits in acquisition and retention of new information, which is learning and memory, showing the strongest correlation with hippocampal-dependent declarative and spatial memory. So the intervention studies, given ethical and practical constraints of investigating brain-related mechanisms in humans, there was a lack of human intervention trials, and 20 animal and 33 human studies, including 16 systematic reviews and meta-analysis, were included. And this is a summary of the preclinical trials. 5 included additional in vitro experiments. 14 of those in vivo studies directly assessed cognitive performance via a variety of tests, and 10 studies additionally assessed BDNF levels, with the majority showing significant improvements, which you can see here with these bold arrows. They show significant findings. This is a brief overview of the findings. So intermittent fasting could significantly increase BDNF and cognition in models of neuroinflammation, Alzheimer's disease, stroke, and in aged rats. It could also upregulate BDNF-induced protein synthesis, dendritic spine density, and long-term potentiation. Intermittent fasting was also linked to the transformation of early to late long-term potentiation, which corresponds to the transformation of early to long-term memory. Recent evidence showed that intermittent fasting could also increase adult hippocampal neurogenesis and differentiation, which was accompanied by improved memory retention and protein levels, and also by improved neurotransmitter signaling and glucose homeostasis. A number of additional pro-cognitive and neuroprotective mechanisms of intermittent fasting were observed and include the regulation of energy homeostasis, reduced oxidative stress, inflammation, and mitochondrial damage. There were some controversies observed. For instance, differential effects of intermittent fasting and caloric restriction on cognition in males and females, and also different BDNF levels in plasma and cortex. This study by Martin et al. Also found significant changes in the endocrine stress response in females, with a complete shutdown of the reproductive system or reproductive hormones in response to energy deprivation, which suggests a higher vulnerability to food scarcity in females. But these results should be interpreted with caution because used a large number of hypotheses without any correction for type 1 errors. The used animal model also seems to impact the response. This study by Lasik et al. Used transgenic female mice overexpressing 5 human familial Alzheimer's disease gene mutations, and they showed increased neurodegeneration and inflammation, as well as a reduced protein synthesis and cognitive performance. But improvements were observed in non-transgenic mice. Their overall study quality was high to acceptable and provided strong evidence for the intervention positively impacting the proposed mechanisms and outcomes. But the heterogeneity of findings can be attributed to a number of factors. There were a lot of different intermittent fasting protocols and duration, a number of different animal models used, and the assessment of cognitive function was via a lot of different tests, which affects different types of memory and learning in different brain regions. Age and gender are a major confounder. Studies are often conducted in young mice, and as most animal studies, mostly male mice are used, which is a big limitation. There are other limitations with animal studies that were identified as well, such as small sample size, gaps in reporting of results, and a large number of hypotheses tested. However, 13 of these studies used suitable post-hoc correction, which increases the reliability of results. So overall, considering the quality, significance, and power of these studies, these consistent findings of animal in vitro studies strongly support the role of intermittent fasting in increasing BDNF levels and cognitive performance by synaptic plasticity and neurogenesis. Human intervention studies. So the systematic search could not identify studies, intervention studies assessing both cognition and BDNF levels. There is strong epidemiological evidence which showed decreased BDNF is associated with cognitive decline and lower brain volume in older adults. And a subgroup analysis showed that decreasing BDNF levels with increasing age and disease progression as well. As mentioned before, the Val66met BDNF gene polymorphism is also associated with decreased memory and executive function. However, the association with higher Alzheimer's disease risk was only observed in women. There were 7 intervention studies investigating the link between intermittent fasting and cognition, and 6 trials measuring the effects on BDNF levels, and which are highlighted here as well. The protocols of intermittent fasting varied greatly, which contributes to inconsistent results. 7 recent studies on intermittent fasting and cognition show promising results. So intermittent fasting and caloric restriction showed improved cognition in healthy overweight or obese subjects, which was accompanied by weight loss. Weight loss was larger in the intermittent fasting group and without adverse effects observed on hunger, dietary restraint, mood, sleep, and quality of life. Similar benefits were observed in a 3-year prospective cohort study in mild cognitive impairment subjects, and that showed additional beneficial changes in metabolic, inflammatory, and oxidative stress markers. The study also did metabolomics, which could distinguish distinct metabolic profiles for fasting individuals, and these patterns were associated with lipids, ketone, and glucose metabolism. There's a lot of research there which you can read up in the paper as well. But just to mention another thing is that this the cross-sectional study by Karin et al. Also identified cognitive benefits of time-restricted feeding for those consuming breakfast, but not for those having dinner, which indicates the importance of circadian regulation. And at this point, I also wanted to mention that we have a very recent publication with Lydia Chambers on the impact of meal timing and nutritional inputs on circadian rhythms. So that plays a role here as well. The 6 studies measuring BDNF levels were of limited quality, mainly due to the study designs and BDNF always being measured as a secondary outcome. So a clinical application, although the scope was limited to one potential pathway among multiple factors in the complex pathology of cognitive decline, the outcome of this research could have useful implications for clinical practice. The lack of human research required the inclusion of animal and observational studies, and they rank lower in the evidence-based medicine hierarchy, but enabled the evaluation of effects on implicated molecular mechanisms, and these were strongly supported in animal studies. Overall, the combined evidence suggests intermittent fasting may be a plausible intervention to upregulate BDNF and support cognitive function in humans. However, the impact of genetics, age, sex, and disease status needs to be considered in a personalized approach. General safety, high adherence, quality of life, and additional system-wide benefits supports intermittent fasting as a promising approach to promote brain health. And, but also, again, optimum duration, protocol, meal timing, and dietary composition require further investigation. There are other potential interventions to upregulate BDNF, mainly exercise and cognitive training, and other possible interventions to improve cognitive function and healthy aging, are, for instance, the Mediterranean diet, omega-3 fatty acids, or polyphenols. So future research, some processes needed to be simplified, and the scope of the project required the exclusion of other lines of inquiry, such as the neuroprotective effects of BDNF, mitochondrial biogenesis, or BDNF upregulation via other peripheral signals. And these are all areas for future mechanism reviews. This research identified a lot of gaps in the literature and raised mechanistic questions, such as the sex-specific stress response, warranting further animal in vitro studies. Some of these areas are listed here and are discussed in more detail in the paper, especially this discussion about the importance of gender. As mentioned before, the effect of intermittent fasting on cognitive function in humans is an understudied area of research and requires high-quality, well-powered human trials. With further progress of research and scientific tools, the integration of the systems biology or functional perspective could allow a more meaningful synthesis of various research disciplines, such as, or including epigenetics, nutrigenomics, neurology, and gerontology, which would all enable practitioners to design more personalized interventions. Yeah, and thank you. That's here are my references, and I welcome any questions.